So hello everyone. Thank you for joining. I know we changed the time of today's seminar, so some of you might have had marked a different time on your calendar. But I hope more people will start to join in the next few minutes. So for those of you who don't know me, I'm Babak Faryabi, Assistant Professor of Pathology, a core member of Penn Epigenetic Institute, and a member of Abramson Family Cancer Research Institute. It's with a great pleasure to introduce Dr. Chao Cheng, who is kindly joining us from Houston today. Dr. Cheng started his scientific journey in China, where he graduated from Fudan University. Then he moved to the US for his doctoral study at USC. Then he left warm California and moved to New Haven for his postdoctoral training in computational biology with Mark Gerstein at the Yale University. After that, during that time, he made seminal contributions to a number of common fund projects, including ENCODE and model organisms ENCODE or mod ENCODE, where he basically studied grammars of transcription factor interaction with the chromatin. After a very successful training, he moved to Dartmouth and became a faculty there in 2012. And a few years ago, 3 years ago, he left Dartmouth for Baylor College of Medicine in Houston, where he's currently an associate professor of medicine and a member of the Institute for Clinical and Translational Research. Dr. Cheng's lab focuses on developing and applying computational methods to advance our understanding of human disease, in particular cancer. In the last couple of years, Dr. Cheng's team have developed a number of seminal tools and methods, and basically pushed the field forward and allowed us to better understand tumor intrinsic and extrinsic features. Today, we hopefully learn about the recent finding and the work from his lab, where we investigated the impact of tumor microenvironment on response to immunotherapy. Without further ado, please join me to welcome Dr. Cheng for our today's Bioinformatics seminar series. Dr. Cheng is all yours. Thank you.
Thank you. Thank you. Okay, let me share my screen.
Yeah, while you are sharing your screen, I just would like to remind everybody, please type in your question into Q&A. So we, if they are very burning, we can answer them during the talk. Otherwise, we wait till end.
Laser. Okay. Hi. Good afternoon, everyone. It's really my pleasure to join this to give a presentation here. And today I will talk about some recent research we have performed that's investigated the association between immunoinfiltration and clinical phenotypes in different types of cancer. And we know that cancer immunology have achieved a big breakthrough in the past 10 years. There are many successful clinical trials that apply immune checkpoint blockade therapy for treating different type of cancer and achieve great success. So one of these example is the Keynote 24, which is a clinical trial that tests the anti-PD-1 therapy in PD-L1 positive non-small cell lung cancer. As shown here, you can see there is a substantial improvement of the progression-free survival in the patient by comparing this immunotherapy treated group with the chemotherapy group. There's a how much there's about 40% of increase about the progression-free survival. And however, if you look at this global view of objective response rate in different cancer therapy, different cancer types, you will see that the majority of cancer types have relatively low treatment objective response rate. It ranges from single digit to 35%. And even in the cancer type that's well known to be responsive to immunotherapy in colorectal cancer, melanoma and lung cancer, the majority of patients are not responsive to the treatment. They don't have durable benefit from the treatment. So I guess in my opinion, like if you want to further improve the checkpoint blockade therapy, we have to address the following questions. The number one, what cancer type are more likely to respond to immune checkpoint blockade therapy? So that means we need to compare in different cancer types to investigate their immunoenvironment to determine which cancer type are more likely to be benefit from immunotherapy. Then the second question is, in a cancer type, which patients are more likely to respond to immune checkpoint blockade therapy? So that's for that questions, we want to develop a biomarker so that you can pre-select the patient that are more likely to be responsive. And these patients are responsive to will benefit from the treatment. And you don't want to, you don't want to treat the patient who will not respond, or you want to find some way, some combination therapy or some other strategy to sensitize the cancer so that they can be more responsive. And the last question is, as I already mentioned, how to sensitize the immune environment so that more patients will be responsive to the immunotherapy. So it will be important to know which pathway have impact on determining the immunohotness in the tumor microenvironment. So this is a tumor immunity cycle. So we start from here. You can see if for some reason the tumor cells are killed, or some of the tumor cells are killed, that will release the tumor cell antigens into the environment. And this antigen will be presented by dendritic cells or other APCs to activate the immune system. For example, they will activate the T cell, and the activated T cells will migrate through the circulation and infiltrated into the tumor microenvironment. And in the tumor environment, they will recognize and attack the tumor cell, and therefore kill the tumor cells. And the killed tumor cell will release the tumor specific antigens to the environment and to initiate another cycle. So here I list there are several features or factors that can determine the tumor microenvironment. The first category of features are genomic features. For example, there's many reports have shown that high level of tumor mutation burden is associated with good responsiveness to immunotherapy. And also the copy number variation, microsatellite instability, and PD-L1 protein level expression are also determined the hotness of the tumor immunity. And another factor is the immunological features, as I'm showing here. And if there's many research shown that the immunocells that are infiltrated into the tumor microenvironment associated with patient response to immunotherapy, associated with the overall prognosis of patient in different cancer type. The third category of factors are environment factors. For example, the microbiota in the digestive system of the patient is associated with the patient response. The virus infection, especially for some virus related tumors, also have strong impact on the tumor microenvironment. The last category of features are genetic features. So that's the factors inherited from the parents, including the HLA homozygosity and the HLA status in the germline cell and the mutation of HLA and their expression in the somatic cells. So that's the last category of features. So accordingly, I will touch three of these feature categories in today's talk. So I will start from the genomic features. So in this part, I will introduce a deep learning based approach for classify tumors into different types, and we will associate this different type to the genomic and clinical phenotypes in different cancer. And in the second part, I will talk about how the immunological feature in the tumor microenvironment correlated with patient clinical outcomes. And lastly, I will briefly talking about how environment factors. And in today's talk, I will only focus on how the impact of virus infection on immunocell infiltration in the tumor microenvironment. Okay, so let me start from the first part. As I mentioned in this part, so we developed a deep learning based algorithm to integrate different types of genomic data for tumor classification. So as shown here, so we utilize 3 types of genomic data from the TCGA. We collected the somatic mutation data of the that's covering more than 5,000 genes across 8,000 patient samples from TCGA. And also we determined 5,000 microsatellite loci in the genome of this patient. And furthermore, we also included the somatic copy number variation information from TCGA that's for this patient. And we're using a multifactorial deep learning model to integrate these features. And at the top of this model, we're using the top layer, which are informative layer, to classify the patient into 4 different categories. So here we choose 4. We find number 1, the 4 is a number that can be easily interpreted. And number 2, we find based on the association with genomic factors, 4 is a number that seems to be most reasonable. And after we determine the 4, we call it the genomic clusters, and we associate with these clusters with genomic features of the cancers, and we associate with these clusters with the immunological features of patient, and also we investigate the difference in prognosis of a patient from different category. So these 8,000 of tumor samples are actually from 29 different solid cancer types. Okay, this is our result. As I said, we based on the genomic feature, we divided the patient into 4 genomic categories: GC1, GC2, GC3, and GC4. And we find that if you correlate this cluster with the global genomic feature, as we're showing here, you can see like GC2 is quite different from the other 4 categories. The GC2 have the significantly high tumor mutation burden, the number of non-synonymous mutations. And the GC2 also associated with the highest level of microsatellite instability. And the GC4 also have high, GC3 have relatively low somatic mutation burden compared with GC4, but higher than GC2 and GC1. And actually, as I said, there are 29 solid tumors have been investigated in this study. So you can see that different cancer types show different pattern of enrichment across the 4 GCs. And like this one, there are 6 cancer types that are enriched in the GC1 category. And there are 3 cancer types, including the endometrial cancer, colorectal cancer, and the stomach cancer, that are enriched in the GC2 category. So that's the one with some specific feature as shown here. And also there are 5 cancer types that are enriched in GC3, and ovarian cancer and breast cancer are enriched in the GC4, which that category is associated with the highest level of copy number variation. And about 50% of cancer there don't have predominant enriched categories. There are distributed in different clusters. I guess the take-home message is GC2 is kind of special, is MSI associated and have associated with highest tumor mutation burden and MSI burden. And actually more than that, even though GC2 is the most special one, this cancer is actually well studied. For example, the colorectal cancer, there are some immune checkpoint blockade therapy have been approved for treating patient with unstable microsatellite instability. But if you look at it more carefully, you will see that the GC1 and the GC2 is also different. So this is more apparent when you look at their immunological features. So here we list the several immunological features at different levels. So first, we calculate the infiltration level of different type of immunocells. And we also select a number of immunostimulatory genes and immunoinhibitory genes. You can see that overall, the GC1 have high level of immunocell infiltration. It has high levels of immuno-related genes in the tumor microenvironment compared with GC3, as shown here. Many of these differences are significant. And specifically, if you look at the PD-1 and the PD-L1, that's the two genes that immuno-oncologists care about. There are significant difference between the GC1 and GC3. Both PD-1 and PD-L1 have significantly high expression level in the GC1 sample than GC3 samples. And accordingly, I mean, GC1 has better prognosis than the GC3. I want to remind that here, these are TCGA patients, so they are not treated by immunotherapy. All these patients are on the standard of care according to the guidelines. But you can see here, GC1, which is the red color group, they have significantly longer survival than the GC3, the blue lines here. And if you look at specific cancer type, so we actually analyze each type, and many of this in many of the cancer type, you see significantly longer survival time of GC1 compared with GC3. And here I just choose two examples. One is the adenocortical carcinoma. The other one is sarcoma. So GC1 is associated with better prognosis on the standard care. Then we want to know how about the responsiveness to immunotherapy. So we collect two data here. One is the Van Allen's cohort, which is a cohort treated by anti-CTLA-4 immunotherapy. And you can see here, when you compare GC1 and GC3, and you will find that the yellow color, which are patients that achieved clinical benefit from the treatment, have high proportion in the GC1 patient compared with the GC3 patient. And this is also supported by the post-treatment survival analysis as shown on the right side. And if you look at another data, basically got similar result. You probably pay attention, notice that in GC2 this time is higher than GC1. But this is probably caused by the relatively small number of samples from the GC2 category. But overall, for the anti-PD-L1 therapy, we also found that GC1 patients are more likely to responsive to immunotherapy than the GC3. Okay, so I quickly introduced the deep learning analysis for cancer type, for cancer classification. Based on this study, we can see that the genomic feature of tumor cells is informative for defining tumor subclasses, and that can tell you which cancer type is more likely to be responsive to immunotherapy. And number two, we find that GC2 is associated with high level of MSI that's supposed to be sensitive to immunotherapy. we know that the GC2 is enriched in colorectal cancer and in endometrial cancers. And GC1 tumors have more active immunoenvironment and are more likely to benefit from immunotherapy compared with the GC3 tumors. Okay, that's basically the part one. And if we move forward, we will investigate how the immuno features correlate with the tumor microenvironment. So when we think about a tumor, in the tumor sample, you have not only the tumor cells, you also have the non-tumor cells. This including stromal cell, different type of immunocells as shown in these figures. And the benefit of this is that when we have a gene expression profile, so the gene expression not only capture the gene expression in the tumor cell, is also informative for the existence of immunocells. That's why, based on this rationale, several deconvolution methods have been developed to infer the relative abundance of immunocell based on gene expression data. So the principle is actually very simple. Let's say if a gene X is only and highly expressed in T cell, and you can infer the abundance of T cell just based on the expression level of gene X, right? So however, we know that, I mean, in the real case, the problem is much more complicated. So there's no perfect marker that can discriminate a specific immunocell. So we have to do something that's more carefully determine the immunocell. So based on this, we utilize the purified immunocell gene expression profiles from purified immunocells to define a gene signature as a reference for inferring the immunocell infiltration in the tumor cell based on the gene expression. And we're using a statistical model called BASE, which is a rank-based method to determine the relevance of immunocell in cancer samples. And this is a figure that gives you a little bit more details. So what we do is, from the imaging project, there are the experiment profile for about 200 different immunocells or hematopoietic cells. And we calculate the specificity of all genes for each of the immunocell types. So the basic idea is, if a gene is more specifically in, say, CD8 T cell, we want to assign weight, because we know that this gene is more informative for determining CD8 T cell levels in the tumor microenvironment based on the tumor gene expression profile. And also, we also take into account the genes that are specifically have low expression levels in a particular immunocell compared with tumor cells, stromal cells, or other immunocells. So we define a pair of signature that capture both upregulated genes and downregulated genes in a specific immunocells. And then we look at, given the tumor gene expression data, we look at the distribution of these genes in the sample, and we're using a rank-based statistics to quantify the biased distribution of immunocell genes highly expressed in the immunocell and genes lowly expressed in a particular immunocells to calculate the score. The score tell you the relative importance or relative abundance of that particular immunocell types. So after developing the method, we first validated this algorithm using data from PBMC from the blood samples. And in that data set, there are collected a collection of individuals with blood samples. They performed RNA-seq experiment to determine the gene expression in each of the patients. They're also using flow cytometry to quantify the abundance of 6 types of immunocells experimentally. So we apply our algorithm to the RNA-seq data to calculate the inferred immunoinfiltration score immunocell abundance and compare with the experiment value as shown here. So overall, there's a relatively high correlation between the inferred value and the experiment value. And if you calculate the pairwise correlation between the two values, you can see that it's relatively quite specific. But you probably also notice that because the two types of B cells, naive B cell and memory B cell, are quite similar in their expression pattern, and you can see there have a high cross correlation between the two types of immunoinfiltration scores. Okay, so that is from blood samples, and we know that the tumor sample is actually quite different from the blood sample. So luckily we find a data that's experimentally determined the immunoinfiltration in a collection of non-small cell lung cancer samples, and there have that experiment data that also provide gene expression data. So we do the inference and we calculate the correlation, and you see that like in the tumor cell for CD8 T cell and B cell, and the correlation is fairly good. So it's okay to be applied to the immuno environment of the tumor samples. And given the tools, now we want to address some clinical relevant questions. So the first one, we want to study how immunocell infiltration correlates with patient survival. So here we use a lung adenocarcinoma dataset as an example. And in the dataset, there are about 250 different lung adenocarcinoma samples. And we calculate the infiltration level of the 6 immuno cell types. And we just simply do the hierarchical clustering using the infiltration score. And you can see overall, there are 2 subcluster of samples. So with on the right side, with relatively high level of immunocell infiltration, and on the left side, with relatively low levels of immunoinfiltration. If you compare the survival, you will see that the yellow group with high level of immunoinfiltration have significantly longer survival compared with the green one, which have low immuno environment. And if you look at each immunocell type individually, you will see that high level of naive B cell infiltration, CD8 T cell infiltration, and CD4 T cell infiltration is associated with longer survival in the lung adenocarcinoma samples. However, the myeloid cell, high level of infiltration of myeloid cell, which is overall, most of these cells are macrophage, they are associated with poor prognosis of the patient. And in this study, we actually pay a lot of attention to the early stage cancer. And you can see here, so when in this cancer, we focus on stage 1 and stage 2, but you can see here, if you adjusting the impact of clinical variables, this immuno signature is also prognostic. So as shown here, we consider actually the gender of the patient, the age of the patient, the tumor stage of the patient, and the smoking status of the patient. After the adjustment, the immunoinfiltration score are still significant. And if you like stratify the patient based on the stage, and you see that in stage 1, naive B cell is also significantly associated with the prognosis. The same story for CD8. And again, like monocyte is associated with poor prognosis in stage 1 cells. This is a figure that we try to stratify the patient by combining the inferred CD8 T cell and the CDR4 gene expression. Remember, we do our inference based on the gene expression data, right? So we can decide the expression level of key immuno checkpoint genes. So here, based on a combination of CDR4 gene expression and the inferred CD8 T cell abundance, we stratify the patient into 4 categories. And interestingly, you can see here, the blue group, that's the group with high level of immunocell infiltration, CD8 T cell infiltration, and low level of CDR4 expression. All these patients are survive without progression. And in contrast, the yellow group, this patient, they have high level of CD8 infiltration. However, they also have high level of CDR4 expression. And you can see here, this patient group have about 25% of patients progress into more aggressive cancer within 5 years, and which is a significant difference from the blue group. And the red group and green group, they have the worst prognosis compared with the other group, because this group, they have low level of CD8 infiltration. Regardless of CDR4 expression level, they always have the worst prognosis. So then we want to know like how the immunocell, we want to extend this analysis to all cancer types. So we perform a pan cancer analysis. We calculate the infiltration level of a number of immunocell types across all the cancer types. You see that so all these different immunocells have quite substantial difference between different cancer types. Like overall, in some well-known cancer type, lung cancer or melanoma, they tend to have a high level of CD8 T cell infiltration, B cell infiltration. But the basic idea is different cancer type vary dramatically in terms of immuno cell infiltration. That's probably relevant to their aggressiveness, to their response rate to immunotherapy. And this figure, we utilize a collection of gene expression data sets, more than 150 different microarray data sets with both gene expression and survival information. So we calculate, we use Cox regression analysis to study the correlation of CD8 T cell with patient prognosis. If for a cancer type, there are multiple data sets can be found. So we perform a meta-analysis to integrate the p-value and the statistics. And you can see here, like in breast cancer, in how many, 9 cancer types, we got evidence that CD8 T cell is protective to the, which is associated with good prognosis. And on the right side, we list 4 cancer types, including glioma, melanoma, bladder cancer, and lung adenocarcinoma, in which CD8 is associated with longer survival of the patient. And we actually also done this for other cancer types. And in 4 cancer types, we detected the association of B cell with prognosis. In 8 cancer types, we find the significant association between NK cells with survival. And for the macrophage, we find in about more than 10, I guess, cancer types, there's association with poor prognosis. And then we want to go to the immunocheckpoint blockade treatment data set. Specifically, we find that it seems that memory B cell signature that's informative for memory B cell infiltration is associated with patient response to immunotherapy. And as shown here, so we first tried the immunotherapy that targeted PD-1 or PD-L1 in the following 2 data, the A and C. And in the Snyder data is an anti-PD-L1 treatment. You can see that if you compare the patient that are benefited from the treatment with the patient that are not benefited from the patient, you will see that the benefited group have significantly higher memory B-like score than the non-benefited group. And this is supported by their survival time. Like the patient with high MBL score have longer survival after the treatment. And also this can be validated by another data set, which is the patient are treated anti-PD-L1. This is a melanoma cohort. And then we know that the mechanism of anti-CDR4 treatment is actually different from the anti-PD-1 or PD-L1. But again, we show that if you stratify patients based on their clinical outcome, there are significant difference in terms of the signature score you can see there is a gradual increase of the MBL score between the three groups. And again, this result is consistent with the post-treatment survival of the patient. And the B cell actually, we know this is a B cell because there are, the signature is correlated with several memory B cell markers. And we published the paper in early 2018. And we were confused at that time because at that time, most of the research focused on the CD8 T cell. But last year, I'm so happy, let's see, there are 3 papers published side by side. And to support that, B cell is actually associated with survival and immunotherapy response. Then we want to see, is there any chance to sensitize a patient that are not immunohot? So then the question is, what pathways have impact on the CD8 T cell infiltration? So we actually perform a very simple analysis. So we have the patient-specific CD8 T cell infiltration level. So we use GSVA analysis to calculate some cancer-related pathways. We calculate the patient-specific pathway score, and we correlated the CD8 T cell infiltration with the pathway activity score. And we find that the MH1 antigen-presenting pathway activity is negatively correlated with CD8 T cell infiltration. I guess this represents a negative selection, which means if a tumor sample have high activity of MHC1 antigen-presenting pathway, these cells are more likely to be eliminated by immunocells. And therefore, you see a negative trend, a negative correlation between them. And there's also positive selection as shown on the right side. The activity of FGFR ligand activation pathway is positively correlated with the CD8 T cell infiltration. So we also have some other data that's giving you some hint that probably by targeting some pathway through combination therapy, you can improve the overall treatment efficacy. Yeah, okay, so I guess now I can summarize the second part as the infiltration, immunoinfiltration are associated with patient prognosis in many cancer types. And there are some immunocell subsets that can be used as biomarkers for predicting patient response to immunotherapy. And at last, we show a memory B-like signature is associated with patient prognosis, associated with patient response rate to immunocheckpoint blockade therapy. Yeah, that's basically the second part. How do we study the immuno features in the tumor microenvironment and how that's relevant to the clinical phenotypes? And finally, I will briefly quickly go through how the environment factors associated with tumor microenvironment. And we know that like about 20% of cancer cases are associated with or caused by virus infection. So especially in liver cancer, the HBV and HCV have high rate of infection. HPV is associated with cervical cancer. EBV is associated with the stomach cancer and several other cancer types. And also there's several other cancer types. We think this is very important because of some clinical trial. As shown here, this is a clinical trial of pembrolizumab in advanced Merkel cell carcinoma, and there are in the clinical trial there stratified the patient in terms of based on the virus status. You can see here, 62% of virus positive patients are benefited from the treatment, but only 44% of virus negative patients are benefit from the treatment. This is interesting because we know that like high level of tumor mutation burden is associated with higher response rate for immunotherapy, right? If you look at the mutation loading of the virus positive and virus negative samples, you will see that the virus negative samples have actually about 100 fold high mutation rate compared with the virus negative. So even though virus positive patient, there are more likely to benefit from this treatment, but this is not definitely triggered by high mutation burden. So virus proteins and maybe some downstream effect of virus infection trigger this difference. So we performed a systematic analysis in 6 different virus-associated cancer types as shown here. So this is some a global picture about the result. And you can see here, we studied the infiltration level of several different types of immunocells between virus positive and virus negative samples. Here, the high, I guess the light color indicate virus negative. The strong color indicates virus positive. You can say overall, the majority of immunocells have higher infiltration levels in virus positive than virus negative samples in each of the cancer type. There are many of them are significant. This is true for both, for all these cancer types. And if you look into more detail and just investigate stratify patient just based on the particular type of virus or strain of virus, you can still see this trend. Like you can see the majority of specific type virus positive samples have high infiltration level than the virus negative samples. And also this also have impact on the T cell infiltration in the environment as shown by the T cell receptor analysis using the RNA-seq data. Especially in stomach cancer, you can see here, the HHV, the EBV positive samples tend to have significantly lower TCR diversity. So that's indicate T cell expansion. Usually a T cell expansion indicates some T cells are selected and therefore imply a potential T cell activation in the samples. And we also merge the data from different cancer types and perform a meta-analysis to see what virus is associated with reduced TCR diversity. You can see like the EBV have significantly reduction in the TCR diversity that's imply a functional T cell environment. And we also studied other features and studied how virus infection have impact on the prognosis. And we find that there's the virus infection have 2 major effects. The number 1 is can change the tumor cell proliferation rate. And number 2 is also alter the immunocell infiltration. So the balance of the 2 effects determines the prognosis of the patient. As I'm showing here, like on the top, I show the head and neck cancer. You can see patient with high level of virus. When the virus loading is high, they are associated with a longer survival. So if you look at the MKI 67 expression, that indicates the proliferation rate of the tumor cells. There's no significant difference. However, as I showed previously, the virus positive sample have significantly high level of overall immunoinfiltration, and accordingly, virus infection is associated with good prognosis. Bladder cancer provide a different example. You can see here, the virus positive sample tend to have poor survival than the virus negative samples, and it turns out that the virus infection will significantly improve the proliferation rate of the tumor cells. And even though it's also increased the overall immunoinfiltration, and in this battle, the tumor cell win the game. So they're associated with the poor survival. So now I would like to summarize my part 3. So the virus positive tumors tend to have high level of immunoinfiltration than the virus negative samples. And the virus infection can increase the proliferation of tumor cells. The balance between proliferation and immunoinfiltration determines prognosis. Yeah, we also have some study that's investigate how the HLA. Remember, in the very beginning, I introduced 4 categories of factors that have impact on the tumor microenvironment, but limited by the time, I deleted that part from my slides. Finally, I would like to thank, give the acknowledgements to all the lab members and collaborators and that are involved in this, contribute to this project, and the funding from NCI and the separate grant. Yeah, I guess that's it. Thank you for your attention. I would happy to take your questions right now.
Thank you, Dr. Cheng. Maybe while waiting for questions to come in, I have a question about, so the markers you had in different cell types in your story, the second story, have you looked those markers or those signatures in abundance of single cell analysis that are currently available to see if they basically corroborate the finding you had using the bulk analysis?
Yeah, we actually try that. We find that the marker actually, they tend to have, let's see, like the genes with high weight for CD8 T cell tend to have significantly high expression levels in the single cell CD8 T cell clusters. We tried, I guess, one is melanoma data. Yeah, there's definitely a consistent, but we also have some surprising results. So some genes is not consistent with the single cell results, maybe due to the dropout rate or some other reasons. And interestingly, there's other things. We find that the genes that are have low expression levels, specifically in an immunosubtype, sometimes also contribute to the final infiltration. So that, yeah, that's something like people have highly overlooked. Yeah, most algorithms just ignore these genes.
Hi, Chao. This is Yixing. So, very nice talk. So I have a question for you. It seems that when you were looking at the immune cell infiltration, you are primarily looking at one tumor type in TCGA, like as a whole cohort. I'm just curious that for certain tumor types with a clear somatic molecular drivers, I wonder if you have grouped the tumor samples into status of somatic drivers and compare the the status of immune cell infiltration across different subgroups.
Yeah, that's a really good good question. Actually, we did that, but we found that so your idea is we regardless of the cancer type, we just collect the sample based on the mutation status of particular gene, right?
So for example, like say certain oncogenes like MYC or some other oncogenes are amplified or overexpressed across many different cancers. So I just wonder if there's some kind of common features of immune cell infiltration.
There do share some common features. Some of the associated with mutation associated with lower abundance of immune cells. But if you quantify the relationship, you will still find that overall, the cancer type, the tissue origin explain the largest proportion of variations of the immunocell variation. So maybe the infiltration is not predominantly determined by a single somatic mutation, right? It's determined by a combination of different factors, the many different mutations, all determined by the overall tumor mutation burden of the patient. Did I answer your question?
Yeah, that's great. Yeah, thank you.
Thanks.
So if there's currently there's no question in the chat, so maybe I ask a second question. I don't want to take all the time, but so as a member of pathology department, I'm wondering if your data can be used to predict or come up with markers of various type of immune cell infiltration, like those kind of immune signatures that people use or reported in previous studies. Have you looked at such possibilities?
So can you give more details about that? So I guess
gene signature that probably can be used as doing a, let's say, PCR-based experiment instead of doing flow to figuring out what are the composition of immune cells within the tumor, but rather using your signatures for more low complex analysis to predict. Let's say you have more B cells or T cells. What subtype of tumor you are dealing with in both stories, your first and second stories. So for instance, your GCs, are there limited number of genes that could say if a tumor belonged to GC1 or GC2, 3, or 4?
Oh, you mean like instead of using the whole gene signature, so can you find a small number of genes that can do the classification, right?
Yes, because
actually, that's a really good question. We got the I mean, when we write the paper, we got the same questions from several different reviewers. We actually tried that. So remember, we have a weight profile, right? So we do some shrinkage analysis. We introduce a shrinkage coefficient to intentionally force some low weighted genes to have a weight of 0, right? And we gradually we tune this shrinkage coefficient to see by cross-validation we can determine can we achieve a best number of genes that give you the best classification result. And we found that actually you can actually got some good classification just using less than 100 genes with the highest weight. Of course, it depends on what immune cell types. I guess NK cell is relatively easier because it's have big difference with other immune cell types and tumor cells. CD4 and CD8 T cell is relatively difficult, so you need more marker genes in the signature.
I see.
Yeah.
Great, thank you. So I think, is there any other question for our speaker? If not,
hi, I've got another question, Dr. Chen. So I may miss some of the earlier part of the talk, but I'm very curious because you get an example of showing head neck with HPV positive cancer showing better prognosis. Do you have another kind of example that with the virus infection, actually the balance shifts to the more rapid, tumor growth, and thereby the total, overall prognosis gets worse?
Oh yeah, we do have like as showing here. Maybe let me zoom in here.
Oh,
so
okay.
head neck cancer, you see that the virus infection associated with good prognosis, right? The upside is the bladder cancer. So high virus loading associated with poor prognosis. And
what is the virus here in bladder?
For this one, the virus. Let me think about the bladder cancer, HPV.
Oh, still HPV. Okay.
HPV, yeah. No. For the head neck is head neck is also HPV. Yes.
Yes. Yeah. But I didn't know that the bladder has a lot of HPV infectious cases.
The proportion is much lower head neck cancer and cervical cancer, but there's still several % of patient tumors have that.
Okay, thank you.
Yeah. All right.
Okay. Any other question for our speaker? Great. Thank you so much, Dr. Cheng, for your time, for a great talk. And I hope you enjoy the rest of your visit.
I did. Thank
Bye.
you very much. Thanks.